Hello friends, hi, good morning. Welcome to our channel, The Voice of Nurse. Today we are going to discuss a continuous topic from the previous videos. The subject name is microbiology. In microbiology, we already finished 3 parts related to the microbiology, FAQs and introduction of the microbiology. In part 1, we discussed related to the introduction, some of the introduction topics and related to the microbiology and the father of microbiology and antiseptic surgery, father of antiseptic surgery and sterilization technique invited by. These all things we already discussed in part 1. In part 2, we finished from the 21 to 50 questions. 30 questions we discussed in part 2 video. Next, part 3 related to the microbiology. In part 3, we continued from the 51 question to 70 question. 30 questions we discussed in part 3 video. Now, today's class we are going to discuss a question number 81 to 100 questions. In this video, we are going to discuss 20 questions in detail. We will see. Before moving on to topic, a small request friends, anybody watching first time my channel, please subscribe my channel, click on the bell icon for further notification. Please share the video to your friends. Let's move on to topic. The question number 81. Best method for getting pure culture is. What is the best method for getting a pure cultures? First we will see the options first. Option A, streak plate. Option B, agar slant. Option C, A and B. Option D, disc plate. Obtaining a pure culture of bacteria is usually accomplished by spreading bacteria on the surface of a solid medium so that a single cell occupies an isolated portion of the agar surface. The single cell will be go through repeated multiplication to produce a visible colony of similar cells or clones. The pure culture techniques methods is streak plate method, micromanipulator method, spread plate method, pour plate method. These 4 techniques are used for the isolating pure cultures. So here the best method for getting pure cultures is option A and B is the correct answer. Streak plate and agar slant methods are used for the getting pure cultures. Agar slant tube and streak plate is the correct answer here. To make it an slant tube, the agar is allowed to cool with the tube laying at an angle of resulting large surface area for spreading a culture. This is the method using a agar slant tube. So here option C is the correct answer. Next we will move on to question number 82. Some dyes are used as. Some of the dyes are used as. We will see here options. Option A, skin and wound antiseptic. Option B, as antibiotics. Option C, A and B. Option D, none of the above. What is the dye? What is called dye? The term dye refers to compounds which can impart color to a substrate when applied in solution from either aqueous or organic solvents. This is called as dye. Some dyes can be used for the skin and wound antiseptic. Which dyes are used antiseptic and disinfectants? Dyes are acriflavine. Proflavin belongs to the category of acridens dye. They are more commonly used as antiseptic and more effective against gram-positive bacteria. The antiseptic agents in dermatologic surgery commonly includes chlorhexidine, providine, iodine, hydrogen peroxide. These all are the antiseptic agents in dermatologic surgery commonly included. They should be used for most, if not all, procedures that enter the dermis of the skin or deeper. So here some dyes are used as. Option A is the correct answer. Skin and wound antiseptic purpose. The some dyes are used for the skin. So here option A is the correct answer. Next we will move on to question number 83. Albert staining is a type of. Albert staining is a which type of staining? We will see here options. First option A, simple staining. Option B, special staining. Option C, differential staining. Option D, none of the above. What is Albert staining? What is Albert technique? We have to know first. Albert staining technique is a type of special staining technique, since it is used to demonstrate a special structure in bacteria. It is chiefly used to demonstrate metachromatic granules found in Corynebacterium diphtheria. This bacterium is responsible for the disease of diphtheria. Albert stains was first described in 1920 and is a type of differential stain used staining the velutin granules, also called as metachromatic granules or food granules, found in Cyanobacterium diphtheriae. So here Albert staining is a special staining. Option B is the correct answer. What is special staining? The special stains are processes that generally employ a dye or chemical that has an affinity for the particular tissue component that is to be demonstrated. They allow the presence or absence of certain cell types, structure and or microorganisms to be viewed by the microscopically. So here option B, Albert staining is a type of special staining. Special staining is the correct answer, option B. Next we will move on to question number 84. Source of infection of fungi like Histoplasma is. The source of infection of fungi like Histoplasma is, we will see here option A water, option B soil, option C animals, option D A and C. The histoplasmosis is an infection caused by fungus called as Histoplasma. The fungus lives in the environment, particularly in the soil that contains large amount of bird or bat droppings. So here we got a answer. Histoplasmosis is infection caused by fungus called as Histoplasma. The fungus lives in the environment, particularly in the soil. So here option B is the correct answer. The histoplasmosis is caused by the reproductive cells or spores of the fungus Histoplasma capsulatum. They float into air when dirt or other materials is disturbed. The fungus thrives in damp soil that's rich in the organic material, especially the droppings from the birds and bats. So here option B, source of infection of fungi like Histoplasma is soil, is the correct answer. Next we will move on to question number 85. Reduction of virulence is known as. What is the called as reduction of virulence? We will see options. Option A exhalation, option B attention, option C invasiveness, option D virulence. We will see one by one what means. Option A exaltation. Exaltation is a feeling or state of extreme happiness. This is nothing but extreme happiness. But here a reduction of virulence is known as. Next option B. Here option B, note attention friends. Attenuation. 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 We will see what means attenuation. Attenuation is the reduction of the force, effect or value of something. The reduction of the amplitude of a single electric current or other oscillation. So here reduction of virulence is known as attenuation. So here option B is the correct answer. Reduction of virulence is known as attenuation. Enhancement of virulence is known as exaltation. So here option B is the correct answer. Next we will move on to question number 86. The formation of a crucial step in anaerobic digestion. The formation of the crucial step in anaerobic digestion is what? We will see here options. Option A exception, option B acetate, option C hydrogen, option D water. What is anaerobic digestion? Anaerobic digestion is a beneficial technology to treat wastewater, sludge, and bio-waste for the production of renewable energy in the form of methane. In this process, here acetate is an important for the methanogenesis. So here formation of crucial step of anaerobic digestion is acetate. Acetogenesis refers to the synthesis of acetate, which includes the formation of acetate by the reduction of carbon dioxide and formation of acetate from the organic acids. The process major steps for the anaerobic digestion. The process of anaerobic digestion takes place through four successive stages like hydrolysis, acidogenesis, acetogenesis and methanogenesis. So here from the options acetogenesis is there means acetone, acetate. Acetate is the correct answer here. Next we will move on to question number 87. The principle involved in the litton air sampler is what principle is involved in the litton air sampler? We will see first options here. Option A filtration, option B electrostatic precipitation, option C impingement, option D all of the above. The microbial air sampler is based on Anderson's principle of Sievin impactor method. The principle that underlies this sampling device is simple and ingenious. Based on the principle is involved in the litton air sampler is electrostatic precipitation. What is meaning of electrostatic precipitation? The electrostatic precipitation is also called as ESP. It can remove particles from a gas stream by using electrical energy to charge particles either positively or negatively. The charged particles are then attracted to collector plates carrying the opposite charge. So here the principle is involved in the litton air sampler is electrostatic precipitation. See here option B is the correct answer. Next we will move on to question number 88. Protein synthesis is inhibited by which of the following antibiotic? The protein synthesis is inhibited by which of the antibiotic from the following? We will see here options. Option A gentamicin, option B kanamycin, option C A and B, option D none of the above. What is protein synthesis? We have to know first. The protein synthesis is the creation of proteins by cells that uses DNA, RNA and various enzymes. It generally includes transcription, translation and post-translation events such as protein folding, modifications and proteolysis, protein synthesis, schematic diagrams. The antibiotics can inhibit protein synthesis by targeting either the 30 subunits. Example for antibiotics for the protein synthesis is streptomycin, tetracycline and aminoglycosides, kanamycin and streptomycin. Gentamicin. These all are the antibiotics can inhibit the protein synthesis. So here protein synthesis is inhibited by which of the following antibiotic means option A and B. Gentamicin, kanamycin is the correct answer. Along with these streptomycin, tetracycline and aminoglycosides and streptomycin. These all are the antibiotic inhibits the protein synthesis. Next we will move on to question number 89. Endospores were first discovered by. The endospores are first discovered by. We will see options. Option A Tyndall, option B Robert Koch, option C Ferdinand Kohn, option D Robert Hooke. What is endospore? An endospore is a dormant, taut and non-reproductive structure protected by certain bacteria from the Firmicute phylum. Endospore formation is usually triggered by lack of nutrients and usually occurs in gram-positive bacteria. In endospore formation, the bacterium divides within its cell wall. The bacterial endospores were first studied 130 years ago by Ferdinand Kohn in year of 1876 and independently by Koch in the same year. Bacterial spores was first studied about endospores by the Kohn. Ferdinand Kohn, a pioneer in the developmental biology of lower plants, considerably promoted the taxonomy and physiology of bacteria, discovered the heat-resistant endospores of bacilli and was active in applied microbiology. Here, endospores were first discovered by Ferdinand Kohn. So, here option C is the correct answer. Next, we will move on to question number 90. Method used for performing the test for sterility or the method are using for performing a test for sterility. We will see here options. First option A, product flush. Option B, membrane filtration. Option C, direct transfer. Option D, all of the above. What means sterility? Sterility is the quality or condition of being sterile. The disease can cause sterility in males. Sterility is the inability. What is meaning of sterility? Why it is called as sterility means the sterility is the inability to produce a biological child, most common in males, while infertility is the inability to conceive after a certain period. Sterility is rarely discussed in clinical literature and often used with a test for the sterility or infertility. Here, the test for sterility are carried out by the method of membrane filtration, by the method of direct transfer, or by addition of concentrated medium to the product. The method of membrane filtration should be used for the method of choice for feasible. So here, the method used for the performing test for sterility is three methods is there in sterility testing or membrane filtration, direct transfer and product flush. So here in these options all of the above is the correct answer. Test for sterility is methods or product flush, membrane filtration and direct transfer. These all are the methods for the test for sterility. So here option D, all of the above is the correct answer. Next we will move on to question number 91. The interval blow exposure to infection and the appearance of the first symptom. So here the interval blow exposure to infection and the appearance of the first symptoms is called as. We will see here options. First option A, late period. Option B, incubation period. Option C, latent period. Option D, A and B. The time elapsed between exposure to a pathogenic organism or a chemical or radiation and when symptoms and signs are first appearance. The term is called as incubation period. The incubation period is the takes for an infection to develop after a person has been exposed to a disease causing organism such as bacteria, virus or fungi. The incubation period ends when the first signs or symptoms of the disease disappear. The incubation period is the time between a person contracting a virus and their symptoms starting to show. For example, for the incubation period of chickenpox is 14 to 16 days. Interval blow exposure to infection and the appearance of first symptoms is the period is called as incubation period. So here option B is the correct answer. Next we will move on to question number 92. The primary differences between the cilia and flagella are. What are the primary differences between the cilia and flagella are? We will see first options. Option A, length and location. Option B, arrangement of microtubules. Option C, number, length and direction of force. Option D, no differences between cilia and flagella. What is cilia? What is flagella? Cilia we will see first. Cilia are small, slender, hair-like structure present on the surface of all mammalian cells. What is flagella? Flagella are microscopic hair-like structure involved in the locomotion of cell. The word flagella means whip. The flagella means the structure involved in the locomotion of the cell. This is a hair-like structure. What is the difference? Primary difference between cilia and flagella are the cilia and flagella are cell organelles that are structurally similar but different in length and function. Cilia are present in organisms such as paramecium, while flagella can be found in bacteria and sperm cell. Cilia are shorter and more numerous than flagella. So here option C is the correct answer. Primary difference between the cilia and flagella is are number, length and direction of force. Next we will move on to question number 93. Photosynthetic eukarya. We will see first options bacteria, option B, protozoa, option C, egale, option D, virus. What is photosynthetic eukarya? The photosynthetic eukarya is defined as a vast assemblage of autotrophs. The emergence dates of these taxa have proven difficult to establish solely on basis of fossil or biomarker evidence. The photosynthetic eukarya can define in the plants and egale. So here the photosynthetic eukarya is present in the egale. In the options, option C is the correct answer. Photosynthesis is plants and egale takes place in chloroplasts and entails two steps, energy transferring and reaction. So here option C is the correct answer, egale. Next we will move on to question number 94. Pyrogens will stimulates. The pyrogens will stimulate which of the following? Option A, kills cell infected with virus. Option B, provokes fever. Option C, kills microorganisms. Option D, A and C. What means pyrogens? Pyrogens is a substance typically produced by a bacterium which produces fever when introduced or released into the blood. A pyrogen is a substance organisms or their product toxins are that provokes fever. Exogenous pyrogens are substances which originate outside the body and which are capable of inducing interleukin. The hypothalamus, which sits at the base of the brain, acts as the body's thermostat. It is triggered by floating biochemical substances called as pyrogens, which flow from sites where the immune system has identified potential trouble to hypothalamus via the bloodstream. So here pyrogens will stimulate or provokes the fever. So here option B is the correct answer. Next we will move on to question number 95. A complication of Campylobacter infection is one of the complication is there for these options. Campylobacter infection complications. Options: option A, ulcerative colitis. Option B, Guillain syndrome. Guillain syndrome means Guillain-Barré syndrome here. Option C, peritonitis. Option D, all of the above. Some people experience complications such as irritable bowel syndrome, temporary paralysis and arthritis in people with weakened immune system such as those with a blood disorders with AIDS or receiving chemotherapy. The Campylobacter occasionally spreads to the bloodstream and causes a life-threatening infection. The infection with Campylobacter jejuni, which causes diarrhea, is one of the most common cause for the GBS, means Guillain-Barré syndrome. About 1 in every 1,000 people with the Campylobacter infection in the state of Guillain-Barré syndrome. Some people will experience the complications such as irritable bowel syndrome, temporary paralysis and arthritis and Guillain-Barré syndrome. So here option B is the correct answer. The complication of Campylobacter infection is Guillain-Barré syndrome. Here option B is the correct answer. Next we will move on to question number 96. What is the protective covering for the genome? What is the protective covering for the genome? We will see here options. Option A, genome. Option B, envelope. Option C, capsid. Option D, folding. The simplest variants consist of two basic components like nucleic acid or single or double-stranded RNA or DNA and a protein coat. So here the simplest variants consist of two basic components. Those are nucleic acid and protein coat. The capsid which functions as a shell to protect the viral genome from nucleus and which during the infection attaches the virion to specific receptors exposed on the prospective host cell. So here in these options, what is the protective covering for the genome means? Here option C is the correct answer, capsid. What does mean capsid? Capsid is a protein shell of a virus enclosing its genetic material. It consists of several oligomeric or repeating structural subunits made up of a protein. Viral capsid or nanometer-sized containers that possess complex mechanical properties and whose main function is to encapsulate viral genome in the one host. So here the protective covering of the genome is capsid is the correct answer. Next we will move on to question number 97. The foundation of immunochemistry were laid down by. The foundation of immunochemistry were laid down by. We will see here options. First option A, Alexander Fleming. Option B, Karl Landsteiner. Option C, Edward Jenner. Option D, Robert Koch. The foundation of immunochemistry by using the electric field, antigen and antibody could be derived into contact, thus reducing the time to test completion. Protein constituents of serum tissue and the allergens could thus separated and characterized by reaction with specific antisera. The father of blood grouping and immunochemistry is Karl Landsteiner. The foundation of immunochemistry was laid down by the Karl Landsteiner. His works there together with that independently done by the Flexner and Lewis laid the foundation of our knowledge of cause for the immunology and poliomyelitis. So, the foundation of immunochemistry was laid down by the Karl Landsteiner. Option B is the correct answer here. Next, we will move on to question number 98. The mutation involving the replacement of one purine by another. The mutation involving the replacement of one purine by another. We will see first options. Option A, transversion. Option B, transition. Option C, frameshift. Option D, A and B. The word mutation means any change in the DNA sequence of cell. Mutation may be caused by mistake during the cell division. What is called in DNA if purine is replaced by a different purine? In case of transversion, purine is replaced by a pyrimidine and vice versa. So, correct answer is transversion. Here, the mutation involving the replacement of one purine by another. Transversion. So, here option A is the correct answer. Transversion mutation, a kind of base substitution in which change in the RNA or DNA sequence included when purine base is substituted by pyrimide base or vice versa. So here in this option, mutation involving the replacement of the one purine by another is option A, transversion is the correct answer. Next we will move on to question number 99. When a patient already suffering from a infection and acquires another infection from another patient is called as. Here the Patient is already suffering from a infection and acquires another infection from the another patient is called as. We will see options. Option A, latent infection. Option B, cross infection. Option C, contagious infection. Option D, direct infection. Cross infection or cross contamination occurs when one person spreads an infection to another either directly or indirectly, is called as cross infection. The transfer of infection, especially to a hospital patient with a different infection or between the different species of animal or plant. What is meant by cross infection? Cross infection occurs when hazardous germs such as bacteria and viruses are transferred from one person to another person. Infection can spread between people, between the pieces of equipment, or within the body. These infection can lead to a sleep problems. So here the patient already suffering from infection acquires another infection from the another patient is called as cross infection. Option B is the correct answer here. Next we will move on to question number 100. Which of the following method of culture is used to make an estimate of viable bacterial count? Which of the following method of culture is used to make estimate the viable bacterial count? We will see first options. Option A, stab method. Option B, pour plate culture method. Option C, sweep plate method. There are two common approaches to inoculating plates for viable counts. The pour plate and the spread plate methods is the common approaches for the viable counts. Although the final inoculation procedure differs between these two methods, they both start with a sterile dilution of the culture. The plate count method or spread plate release the bacteria growing a colony on nutrient medium. The colony becomes visible to the naked eye and removable of colonies and a plate can be counted. So here in these options, pour plate culture method, sweep plate method. So here option B and C is the correct answer. This is last question in this class, today's class. And regarding microbiology, we are finished 100 questions related to the FAQs in previous year's exams. This is the last question in this video, part 4 related to the microbiology. Thank you so much for watching my video. Next subject we will discuss community health nursing. Stay tuned friends. Thank you so much for watching. And anybody watching first time my channel, please subscribe my channel, click on the bell icon for further notification, and please give a like and share the video to your friends. Thank you friends. Please give me a like.